All right, so next topic is about matched pair procedures. Okay, so we haven't talked about we haven't talked about like different experiments in quite a while. So let's do a little bit of a review about like what matched pair procedures are. But before we jump into that, so comparative studies where we're looking at two different things or like two different people or two different treatments kind of thing are more convincing than a single sample investigation. Okay, so for that sample, a one sample inference is less common than this comparative inference because we are comparing two different things. Okay, we're comparing maybe it's like two almost identical individuals, and one gets one sample or one gets one uh, 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 like one treatment and the other gets the other treatment, or it's the same individual getting both treatments to see which one does better. Okay, so uh, these study designs will result in what we call paired data. Okay, all right. So now let's do a quick review about matched pair. So a matched pair design, matched pair design is a special case of randomized block design. Remember we talked about these block designs a long time ago. All right, so using a blocking variable, subjects are arranged in pairs matched on relevant factors. Sorry, not data, relevant factors. Don't know where that came from. Yeah, relevant factors. Okay. All right, these matched pairs may be formed naturally or by the experimenter. So think we want to get people like as identical as possible so that when we're looking at the differences in treatments, then we can say that it's due to the treatments and not some random, like random factor. Okay. So if you're doing pairs, every pair will receive both treatments. Okay, by randomly assigning one treatment to one member of the pair and subsequently assigning the remaining treatment to the second member of the pair. So they're not, both people aren't getting both treatments. Okay, every pair is getting both treatments. One gets one gets another. Okay, and then they're going to compare at the end. Alternatively, if you're not pairing up like identical or as close to identical as close to identical as possible individuals, you can also just use one subject and give them both treatments. Okay, but the order that that subject gets the treatment is randomly assigned. So remember, when we're talking conditions, we have to look out for that kind of thing. Okay. All right. So paired data comes from measuring the same quantitative variable twice, like either on the same subject or individual. Okay, and we can find the differences between the two data points for each subject, and then analyze those differences. Okay, so we're going to be doing the exact same sort of test we did at the end of the last lesson, but I know, and that was called like a one sample test. Okay, but we're still using a one sample T procedure. Okay. But I know you're thinking, okay, well, we've got two samples, though. We've got the two individuals, or we've got the same individual getting the two different treatments. So why is this a one sample T procedure? Well, it's because we're not looking at both samples like individually, okay? We are looking at the difference in the samples. So ultimately, we still end up with one sample. It's the difference, okay, the mean difference of our group, okay? And this method is called the matched pair, if I could spell, pair procedure. Okay, so we still have the same four steps that we've seen in a while. Okay, all right, we still have to communicate, and it's still a significance test, so it's just the same as it's always going to be, but we're going to be using this mu, all right, so it's mu of the difference. Okay, so mu d. All right. So just go ahead and read through these. There's nothing to hold. There's nothing really different about this piece. But go ahead and like read through that. And the only real sort of difference that we got to remember is if we look at our conditions. Okay, you might notice there's only two. Okay, there's no independence check. And that's because, remember, independence only works with or it's only necessary when sampling. We're doing an experiment, so the independence part of this, the part of the equation is not required. Okay, So it's going to be the end of this video. The next video is going to be the example.